the meeting, and yeah, while Dr. Wenda preparing the
I cannot share because it says host
disabled. I forgot. Yeah, I have to open first. So please. Now it should be able. Yeah, and very good morning, all students and also other participants to environmental toxicology class. So today we are very fortunate to welcome back again Dr. Wenda to deliver the lectures. So this week is already the lecture 4, yeah, and specifically Dr. Wenda will deliver the topic about toxicokinetic and toxicodynamic. Last week he already presented to us the introductions, yeah, regarding to the basic principle in toxicokinetic and also toxicodynamic. And today, I guess the lecture will be further go deeply, yeah, and more comprehensive regarding to the toxicokinetic and toxicodynamic. So I guess without any further ado, Dr. Wenda, time is yours.
Okay, thank you, Dr. Fitri. So yeah, so today we want to make a little bit change up. Initially, the topics for today is different, right? We're supposed to go to we're supposed to go to exposure routes and mode of toxic action, but I made some changes because I think the toxicokinetics and toxicodynamics is not easy to cover within 1.5 hours, right? So I make it into 2 lectures. And we will talk about these exposure routes and mode of toxic action coming next week. And yeah, next week we'll talk about more chemistry, right? Before this, we already look into some introduction. We talk about what exactly is environmental toxicology and why we need to study it. And then we look a bit into the dose-response relationship, which is the main, very important thing in environmental toxicology. And today, this week, we are going to continue from what we left out last week. And before that, just to give everyone some overview of what to expect in terms of the assessment. Okay, so week 4, which is this week, right? This week, we will have a cognitive-based assessment, which means that you need to answer some questions, right? On a piece of paper, maybe like A4 papers, right? And then you need to submit it to a link. Right, the link can be found here. If you go to this website, right? Yeah, you go to this website, then you can see that there's one tab on this KI510 environment toxicology on the assignment. So you can download this assignment and then start doing. The due date for this assignment will be next week on the 7th October. Right, so you have 1 week time to solve all those questions. The question is very simple, short questions. First 8 questions will be like, I ask and you answer in a very short way, right? Maybe 2-3 sentences. And if you need to draw, then you just draw some very simple thing. But then there's also the part B. Part B is more on questions that is like more challenging, right? You need to do some research to solve part B. So one example would be like I asked about arsenic, right? Arsenic as one of the toxic substance, right? Toxic heavy metal. So you need to look online, okay? Go and find out what is the sources of arsenic, and then what is the toxicology effects, okay? Toxicosis of arsenic, right? You need to find out all these things, and then after that you need to write the reference, and then write what is the problem with the data and so on. Right. So have a look at it once the lecture is over, so that you can look at the questions and understand what you need to do. Right. And if you have any questions regarding the assignment, just email me. I'm free to answer. So this week will be cognitive based, right? Week 4, and then week 5 next week we will have a presentation assignment, which means that I'll give you a presentation assignment with the questions, right? You need to prepare. Right? You need to prepare something, right? A video or PowerPoint, and send it. It's not going to be live, but it's going to be like a recorded presentation. So yeah, we will look into the communication and understanding. Okay, and then week 8, the last week, right, we will have a more cognitive-based assessment. This will be something that's more difficult. more towards problem solving. So we'll look into that once we reach week 8, right? Okay, just a short recap of last week's lecture. We talked about exposure phase, right? How a person can be exposed to a chemical, how we can actually look into whether the exposure is significant or the exposure is like not going to affect us, right? So we also look into toxicokinetic phase, particularly the first two, absorption and distribution, whereby we look into how a toxicant can enter into our body through various ways. For example, respiratory routes. We have the dermal routes, whereby toxicant can be absorbed by our skin, and then another one is the ingestion. That means we eat something that is toxic. And out of these 3 ways where toxicant can enter into our body, 2 ways offers direct contact with our blood, which is the respiratory route, whereby we breathe in something, it goes to our lung, it goes to our blood. The other one would be the dermal, right? Dermal, which means if we are in contact with a toxicant, this toxicant can actually enter into our blood directly through our skin, right? And the other one would be ingestion. Ingestion would mean that our intestine absorbs some of the toxicant, and usually if absorption through intestine, it goes to our liver first, right? So it goes to our liver, and then you have certain detoxification effect. Okay? And after absorption, okay, after you absorb those toxicants, next thing will be the distribution. Okay, the toxicant will be transferred to all over our body, and there are many ways whereby distribution can happen. For example, it's through blood. Okay, blood is the most important distribution channel. Okay, whereby most of the distribution happens via our blood. Okay, because it is a pressurized system, it goes to everywhere in our body, right? And then we have the other fluids, other body fluids, like which can also contribute to distribution of the toxicant to target organ. Okay, so today will be 3 things. One is metabolism. We were going to look into more details in the metabolism part, and we also want to look into excretion. Excretion as one of the toxicokinetic phase, right? And finally, we want to look into the toxicodynamic phase, right? To see what kind of toxic effect that a toxicant can manifest, right? Okay, some outline of what we are going to talk about today is role of biotransformation in toxicokinetics, then how biotransformation can eliminate toxicant. Okay, we want to summarize the role of elimination in toxicokinetics. Then we want to look into what exactly is target organ toxicity, which is related to toxicodynamics. And finally, we want to explain a bit on what is the basis for specificity of organ toxicity. Right? All these are the outline that we are going to go through today. Okay, so the third things that can happen will be metabolism. So once you have absorption, then distribution, right? The next thing is that we need to try to remove the toxicant from our body, right? So one way to achieve this is to go through the metabolism phase, right? So generally, metabolism phase is a sum of biochemical reaction occurring to a molecule within the body. Okay, so most of the time, metabolism phase resulted in energy production. So we want to produce energy from this metabolism, right? We heard a lot of this term, metabolism, right? So generally, metabolism depends on many factors. For example, our genetic, right? Last week we have questions on this metabolism. One of the reasons for metabolism to be good or bad, it depends on the genetic factor. The other one is the biochemical factor of our body. So which means that if you have certain hormones, sometimes you can speed up the metabolism rate. One example would be the thyroid hormone. Thyroid hormone is very important for metabolism. So if you have very low thyroid hormone, then of course you have a poor metabolism. right? So generally, this metabolism can happen via two ways, right? There are two important terms. One will be the anabolism. Okay, anabolism means that you are building up, right? Building up means that it is like constructing something from a smaller molecule into something bigger, right? So usually this kind of process, right, needs energy. We need some energy to build those proteins, to combine proteins together, right? The other one will be catabolism. Right? Catabolism means breakdown. We are actually breaking down something, right? Something bigger, like proteins, right, into something that is small, right? Smaller molecules. So for this process to happen, right, breaking down, right, you actually have released those energies. These are the two fundamental processes, right? Generally, these two processes occurs in the cytoplasm, okay, or what we call as the fluids inside the cell, right? It's like, you know, the cell, right? It has a lot of organelles like nucleus, and then you have mitochondria and so on. So inside the fluids, this anabolism and catabolism reaction will happen. Or in other words, we have metabolism, right? And usually the main organ that facilitate this metabolism will be the liver. Okay, our liver is a powerhouse for metabolism to happen. And sometimes, of course, storage itself can affect our body's ability to biotransform and eliminate, right? Storage means that, like, for example, you are ingesting like lead, right? Lead into your body, right? Sometimes this lead itself can be stored in your bone, right? So if it is stored in your bone, this means that that particular heavy metals will not be available for reaction by the body, right? It's stored there via certain bonds or maybe certain reaction, right? It is there, right? And there's no access of our organs to that particular heavy metal anymore, right? So our body will not be able to remove it via various other means, right? So it will stay in our body. Okay, if it's stored in our body, it will stay in our body until a day whereby we have certain certain maybe signals, right? For example, if you are having like dioxin stored in your fats, right? And we are sick and then we lose a lot of weight, right? So when we lose a lot of weight, then all this dioxin from our fats will come out from the lipids, okay, from our fats. So this will cause more effects. And unfortunately, our body, as I mentioned, cannot eliminate. Okay, so why we need biotransformation? A very simple reason is because we want to convert a toxicant. If we talk about the toxicant, we want to convert it from hydrophobic state into hydrophilic state, which means that we want to try to make that toxicant to become more water soluble, right? In other words, there's a term called grease to salt. Grease to salt means that we want to convert something that is very greasy, okay, something that is not soluble, something like this, into something that is water soluble, right? Or hydrophilic compound. Okay, so hydrophilic molecules, the reason why you want to have that is because they are less able to cross cellular membranes, right? Which means that if it's in our blood, right, we can filter it through our kidney, right? It cannot be in our cell so effectively. So this is one of the major reasons, right? And then, of course, the major elimination route, of course, is the feces and the urine. Okay? If it's soluble, it can be easily removed through this urine, okay, as a liquid state, okay? And then feces, which means that through fecal waste, we go to the toilet, and then after that we remove it, okay, from our body. So one of the factors that affects the removal would be the half-life, right? The half-life means that how long it will stay in our body. Okay, that means, let's say we ingest 1 gram of the of a toxicant, right? So half-life means that the value itself means that how long it will take for this 1 gram to become half of the original dosage, right? So this means that we want to know how long it will take. We can actually calculate for it to be removed from our body, right? So this also allows us to compare the rate of removal of a toxicant, or what we call as the depuration rate. Usually we say it as depuration rate, which means our body actually can remove this kind of toxicant, and there's a half-life, a biological half-life in it. And for this biotransformation reaction to happen, there are two phases. Okay, we have phase 1, whereby we consider this as functional group modification. And we have phase 2, which is what we consider as conjugation reaction. Functional group modification in phase 1 means that it is like we want to modify it, we want to oxidize it, okay, to have more oxygen group, to have more water-soluble functional groups. Whereby phase 2, we talk about how we can actually combine 2 molecules, right? For example, you have a toxicant, you want to add in more sugar group, you want to add in more sulfate group, right? And then when we add in all these group, it becomes more water soluble. So this is the goal to produce water soluble metabolites. The term metabolites here means that we want intermediates. It means intermediates, right? Like for example, we have a phenol, right? A phenol molecule, that's the toxicant. And then this phenol can be oxidized by our body, maybe to become something that has a lot of OH group, right? So this oxidized product, we call it as the metabolites, right? So for a toxicant, right, we can have more than 1 metabolites, okay? Because there are many pathways. And one toxicant can become like 5, maybe 10, maybe 100 metabolites, right? It depends on what kind of toxicant that you have, and how many enzymes that is capable of transforming that toxicant into something else, right? So another one is activate natural and endogenous compounds for normal function, which means that we want to activate our certain chemicals, right? So that our body can actually use the chemicals for other things, like other functions, right? So one example would be, for example, if you eat certain drugs, right, that has beneficial value to our body. So sometimes you eat it, and then our body can actually transform it to some other forms, and these other forms can be more effective in that treating that disease. Okay. Some compound, of course, undergo certain reaction that we call as bioactivation, okay? Which means that you have the toxicant, it can be transformed into a metabolite that is more toxic than original compound. Okay, this is this, right, is an example would be like a benzene, right? Okay, benzene is you know that benzene ring, right? Okay, benzene is actually last time it was not regarded as carcinogenic, right? And then it's actually now regarded as carcinogenic, okay, which means it's cancer causing. So one of the pathway that we know is that this benzene itself can actually be oxidized into epoxide, okay, having an epoxide functional group, which make it become more toxic, right? That's the pathway of carcinogenic action by benzene, okay, according to several research, right? Okay, the results of biotransformation would be, one is the increased toxicity. This is the undesirable effect of biotransformation. The other one will be decreased toxicity. Okay, this is the desirable effect. If we want to make it into less toxic. Sometimes you can have no effect. Okay, no effect means that nothing wrong with it. You have a toxicant, you have metabolism, then there's nothing changed. Okay, it's still toxic. There's no effect. Last one will be present to metabolize endogenous compound. Okay, we want it to be, sometimes we want it to be able to carry out certain reaction, okay, in our body, right? So just to show you the transformation, you can see in this diagram, xenobiotics, which stand for man-made chemicals. It can be undergoing metabolic transformation to become primary metabolite. Sometimes it can be transformed into something called the reactive intermediates. Okay, depending on what kind of xenobiotics you have. Okay, sometimes you can have something that can undergo redox reaction, and then it may result in reactive intermediates, right? And you can also have some reaction, okay, phase 1, phase 2 reaction to become final metabolite, of which some of these, okay, primary metabolites, final metabolites, and reactive intermediates may interact with a cellular component, which result in the toxic endpoint. Okay, it gives you some effects due to the biotransformation itself. Okay, two different categories, as I mentioned. We have phase 1 and phase 2. So phase 1 will be governed by several reactions. For example, oxidation, you have reduction, you have hydrolysis, right? These reactions, oxidation, reduction, hydrolysis, they are basically governed by the Gibbs free energy and thermodynamics and also kinetics. Okay, which means that they can happen solely due to the thermodynamics itself, right? So if you have like organic chemistry, whereby you learn about how functional groups can actually react with certain groups, right? And maybe like addition of H2O2 can actually oxidize certain groups like alkene, alkyne, all these groups, right? You can actually predict what kind of reaction can take place, right? Because all these, they are basically based on the thermodynamics itself. And then we have the phase 2 reaction, right? Phase 2 reaction will be more into conjugation, synthesis, addition of certain molecules into the parent molecule itself. Okay, we add certain groups into that molecules. And phase 2 usually most of the time is due to enzyme reaction, right? Yeah, the main goal will be to make it into polar or very polar molecules so that we can actually eliminate it easily. Okay, when we talk about biotransformation in phase 1, okay, phase 1, most of the time is also by enzyme. Okay, we have oxidation, reduction, and hydrolysis. Right, oxidation would mean that we want to make it into a molecule that contains a lot of oxygen group. Okay, we oxidize it, we increase the valence, we increase the oxidation state, make it into something that can be easily soluble. And usually this oxidation can be performed by certain enzymes like cyclochrome P450. This is very important enzyme. You will see this a lot in this biotransformation. And then you have MFO, you have alcohol dehydrogenase. Alcohol dehydrogenase means that it is something that can detoxify alcohol. Oxidase, you have oxidase, and you have others, right? So usually you see this, okay, ASE here means that it is an enzyme. Okay. Then you have a reduction, okay, reduction reaction, which means removal of oxygen, hydrogen, or add hydrogen, and sometimes decrease valence oxidation state, can be conducted by this reductase, right? This one is less important. Sometimes this reduction reaction will lead to this toxic product, right? This reduction reaction has more tendency to lead to something that is more toxic compared to oxidation, right? Then we have hydrolysis, which means addition of water, or maybe a reaction that has water, okay, facilitating the breakdown, right? So you have an esterase, phosphatase, and others. All these are their enzymes. You can see ASE here, ASE here. These are the enzymes that promotes this hydrolysis reaction. Okay, so we have phase 1 reaction. All this can happen. You can see most of the time it is oxidation, right? Oxidation from aniline to become aminophenol, right? And then you have the alkylation, all this all involve oxygen itself, right? And because reduction, we have this reduction reaction. This reduction of azo bond, you can see here. Okay, this azo bond is found a lot in the azo dye, right? The dye that we use to color our clothes. It has this azo bond, and usually this azo bond can be broken down. It can be reduced to NH2, right? So from this example, you can see that azobenzene is like a relatively less toxic compared to aniline. Okay, usually this aniline is amine compound is toxic, right? So we have also hydrolysis, okay, as a phase 1 reaction, right? And a phase 2 reaction usually involve addition of a molecule, okay, like for example, phenyl acetic acid. You have addition of this glutamine amino acids, you will have a larger molecule, right? Same thing for benzoic acid, phenol, right? You have an addition of these large functional groups, sometimes through this bond, O bond, right, and so on. So there are many different kind of bonds. So all these reactions, they are facilitated by enzyme, right? Okay, so in a phase 2 reaction, you have conjugation reaction, and for this conjugation reaction to work, right, usually you have an enzyme plus with a cofactor, right? Cofactor means that you have to have something to add the functional group. For example, let's say you have an enzyme here, right? This is your enzyme, right? Then for reaction to happen, okay, for you to add the functional groups, right? So first, a cofactor has to come in. Okay, let's say this cofactor has a, for example, carbon. Okay, yeah, let's say it has a carbon group here that you CX, right? This carbon group that you want to attach to the toxicant. Then this cofactor will come to the enzyme, and then after that, you have another this toxicant molecule come to the enzyme itself, and then after that, you have a product consisting of enzyme plus with toxicant with the CX functional group plus with the cofactor that has lost the CX functional groups, right? So usually these are the requirement for biotransformation in phase 2 to happen. So enzyme itself acts as a platform for catalysis to happen, right? It does not perform or react that changes its structure, right? That's the definition of enzyme. And enzyme usually can be reused many times, right? So cofactor, on the other hand, donates the group, right? You have to have something that can donate certain groups, right? For example, glucuronic acids, glutathiones, sulfate, acetyl groups, methyl groups, all this needs to be in the cofactor, right? So this cofactor will donate that group, and then after that, your toxicant will accept that group, okay? So all this happens on the enzyme itself, right? So generally, it tends to increase the molecular size. Okay, so when we accept something, of course, the molecular size will increase, right? And the polarity for excretion will also be higher, right? It is more polar. It is more easy to remove it through urine. Some example of cofactors, you have GSH. Okay? If you look at this molecule, it looks very complicated, but then of course, this is the part whereby the reaction happens, right? For acetyl coenzyme A, another cofactor, phase 2 cofactor, it releases this acetyl group. Okay, so this molecule will attach to an enzyme, and then this group here, acetyl group, will be released, okay, and attached to the toxicant. Then we have this PEPs. Okay, this is also one of the important cofactor, right, which release this group. Okay, SO3 group, and then the other one is uridine diphosphogluconic acid, whereby it releases this. This is sugar molecule, right? It releases a sugar molecule, right? So all these cofactors, they have specific function, which is to release certain molecules to your toxicant, okay, by adding certain groups. So one example, benzene metabolism. as I mentioned just now. So benzene itself, we can actually be exposed to this, right? This is benzene, and then this kind of molecules goes in our body, and many things can happen, okay, due to our enzyme, right? Usually the first thing that happens would be the epoxidation, right? Epoxidation introduce this oxygen group here, right, through this enzyme P450, right? So this is a phase 1 reaction, right? And then from here you can have additional reaction, okay, make it into phenol. Okay. This is also a phase 1 reaction, and this phenol itself can be transformed, okay, through phase 2 reaction to become glucuronate, or maybe the addition of this SO3 group through PAPS, right, as the cofactor, right? And you can see, right, you have two different products here, okay, and then the toxic epoxide can actually be transformed further into dihydrodeal, right? So it can also be transformed into this molecule through a phase 2 reaction, right? So there are many different kind of reaction that can happen, which means that one benzene itself, you observe like 1, 2, 3, 4, 5, 6 different metabolites, right? So all these 6 metabolites will be in the body. Okay, so usually in the clinical studies, right, when they want to look into the this metabolism of certain chemicals, what they'll do is that they'll inject these chemicals into animals, right? And then they'll collect the blood. So they'll take the blood and then they will analyze through various approach. For example, they can use LCMS, they can use GCMS, right? Then they will look into what kind of molecules that can be present before introduction of this benzene to the molecules, to the animals, and after introduction. Then they will see, okay, and then they will identify which kind of chemicals that is present after exposure, and from there they will determine, okay, the pathway whereby the toxicant can be metabolized, right? So this example, this benzene example, generally leads from less toxic molecule to carcinogenic molecule. This toxic epoxide is actually carcinogenic. This is the pathway that many people thought benzene carcinogenic activity comes from. Okay. So we have other reactions. Aniline can actually be oxidized through phase 1 reaction, right? And after that, it goes to the phase 2. Then we have this is dimethylpropylamine. It can also undergo phase 1 reaction. Okay, and then after that, this one of the intermediate can go to the phase 2 reaction. And then we have another reaction that is undesirable. Okay, something that we don't really like, which would be the reduction. Okay, reduction of chemicals. For example, this tetrachloromethane, which is reduced to toxic free radicals. Okay, so these toxic free radicals is something that can cause what we call as the oxidative stress to the human body, right? You cannot have a lot of toxic free radicals in your body because this toxic free radical is very reactive. Okay, it can attack everything, right? Your DNA, your cell, your membrane, right? All these organelles in your cell, right, or your body, right, and any organs, DNA, can be attacked by this toxic free radical very easily. Okay, because toxic free radical has a lot of this reactive group. It has a radical here. It has an electron here, which is like it wants to find another partner, right? So it is very reactive and can cause damage to everything, enzyme, proteins, and so on. So as you can see, right, this P450 is not specific, right? It is able to be used to carry out phase 1 reaction to many different kind of molecules, right? So this is one of the reasons why we can actually detoxify a lot of different kind of toxicant, right? Okay, one case study, okay, would be this fluorocitrate and kangaroos, right? This fluorocitrate is actually a toxicant found in plant. Okay, it's actually a natural product, and it is quite toxic. Okay, it is very toxic, and it is found in Western Australia. Right, this toxicant fluorocitrate is actually found in Western Australia, which can affect the certain biochemical reaction, phase 1, phase 2 reaction in our body. Right, so it is found in this plant, gastrolobium and oxylobium. Okay? These two different plants, and it is very lethal. Okay, this is actually quite toxic, and there's this red and gray kangaroos in Western Australia. Right now, they have evolved certain resistance to this kind of toxic natural product. But of course, there was a case, right, many years ago, whereby they are not aware of this kind of resistance, right? So what happens is that sometimes you have animal relocation, right? Sometimes you have certain kangaroos like breeding excessively in a certain part of Australia, and it cannot like support all the population. Okay, that particular area has limited food, so human intervention. Right, we take the kangaroo, we put it in other places, right? Because we want to make sure that the kangaroo can actually survive. So we take them and then we put it in a place whereby you will have this fluorocitrate problem, right? So this is an issue because those rats and great kangaroos in Western Australia has already evolved resistance, but then those been transferred to that particular area. Okay, maybe, let's say northern part, they take this kangaroo and then they put it in the western part, right? The kangaroos in the northern part has no resistance. So what happens is that they learn some lessons, right? So most of the kangaroo from the northern part suffers from certain effects due to these chemicals, fluorocitrate. So this fluorocitrate can interfere with certain reaction in the body, okay, which will affect the metabolism, right? Metabolism of the kangaroo. So of course, from the fluorocitrate experience, of course, we can actually design certain chemicals to make it into rodenticides, which means that we use this chemical. We use the fluorocitrate basis, the biochemical reaction, to design something, rodenticide, to remove those rodents, right? So what happens is that we can actually start with fluoroacetic acid, right? We take this fluoroacetic acid, let's say we have an area whereby rodents infest that particular area. We take fluoroacetic acid, we mix with the food, and then we pass it to them, and then they will suffer from certain effect. Okay? We can actually remove them from the that area, right? So this fluoroacetate can be transformed into something called the fluorocitrate, okay, through a coenzyme, this enzyme, right? So when we have this fluoroacetyl CoA, we actually can create the fluorocitrate metabolites, which can actually affect the Krebs cycle. Okay, Krebs cycle is important for mitochondrial energy production, right? So this is one of the bases that we can actually use to create a rodenticides. Okay, other kind of chemicals, okay, in phase 1, phase 2, that can be toxic will be vomitoxin, okay, and then we have aflatoxin B1 and benzopyrene. Okay, this benzopyrene is actually a chemicals used for found in this high temperature reaction. Okay, for example, when you barbecue certain meats and you see that there's a black color areas, okay, which comes from the burning of the meat, right? That particular area may contain this kind of chemical, benzopyrene. Okay, that's why I don't know whether you have this experience when you barbecue. Sometimes older people, okay, or maybe some people who have more experience, they will say that don't eat the black part, right? The darker part of the meat, right? So one of the reasons would be because that particular black part okay, the one that has been burned excessively, right, may contain certain chemicals like benzopyrene. And this benzopyrene, we call it as carcinogen, right? It can cause cancer, right? So if you are exposed to this kind of chemical every day, then it could cause cancer to the body. Okay, it can damage DNA and so on, right? And aside from barbecue, you also find it in like incineration plant. Okay, like for example, waste to energy plant, a plant whereby we burn coal to produce energy, right? Because we burn at excessive temperature, this benzopyrene can actually be produced. So we have to make sure that the benzopyrene is limited, right? So vomitoxin and aflatoxin, these two are actually coming from nature, right? Nature means that they are found, okay, in plants, in molds, right? So they can actually go into our body and cause effects, okay, toxic effect. For example, aflatoxin, right? This is something that also can cause cancer, right? This aflatoxin is found in like things like corn and peanuts. So when you have this peanut butter, right? Okay, when you go and buy, let's say, a peanut butter from a supermarket, right? You need to be careful, right? Don't buy those that is like not prepared properly, because sometimes they could contain these certain chemicals like aflatoxin, right? If you are not produce the peanut butter properly, if you have aflatoxin, it can cause cancer cells. That's why most of the countries, right, they have these rules saying that for all the peanut butters that's sold, they have to be tested for this aflatoxin, right? To prevent unwanted problems, right, like cancers. Okay, another one example, just to share with you, is that this heavy metal toxicity. Okay, let's look at the case whereby we have lead, okay, as one of our target heavy metals. So we want to see how we can actually remove lead from our body and how we can actually treat lead. Right? So lead itself is readily absorbed. Okay, in terms of absorption, it can be absorbed by our calcium channels as divalent ions. Okay, so lead itself is something that can cause adverse effect to our body, right? And it depends on what kind of species that you have. Whether you absorb metallic lead into your body, or maybe absorb the salt. Okay, the salt is more toxic because it's readily available, it's very mobile, right? So it depends on the species that you have, that you absorb, right? So it is capable of react with many binding sites because of the nature of lead. Okay, this is a heavy metal, right? Lead is a heavy metal which can bind with a lot of ligands, right? So our protein in our body contains a lot of functional groups that can act as ligands. So this is the reason why lead can actually affect our body, because it has this reaction to ligands, right? So one of the important functional group with it will be this sulfonyl functional groups, which are present in many of these important enzymes, right? So if you have lead, this lead will bind with this kind of group, right? It can also bind with amino group, phosphate group, imidazole group, hydroxyl group in the enzymes and proteins. In terms of these heavy metals, we know, okay, absorption can happen through indigestion. It can happen through respiration. Digestion means that perhaps we eat something that contains lead. Respiration can happen. Sometimes we breathe in the lead dust, right? Sometimes dust can contain a lot of things, right? So when we breathe in that, it goes to our blood, right? And in terms of detoxification, we have our liver, right? Liver can, to some extent, function as a site whereby it can detoxify certain lead, okay, lead compound, right? So we have kidney, which can help in excretion. Okay? Lead is soluble, so we can excrete it through our urine. And of course, because we know that it is a ligand, right? And it is something that can bind with a ligand, right? So antidotes will be competing ligands, right? Which means that, let's say you have a receptor, right? Then you have this lead that can bind with this receptor, right? So we add in certain competing ligand like EDTA, right? So we want this lead to bind with the EDTA instead of the receptor that you have. So we can ingest this EDTA in our body, right? The safe grade one, not the chemical that is found in your lab, right? You need to find the safe EDTA, right? The one that is for drug, okay, for human consumption. You eat it, and then this EDTA will be able to compete with the receptor to bind with the lead itself, and then this will result in lead and the EDTA binding. We call it as a complex, and we can actually remove it through urine. Okay. So in terms of absorption, metallic lead absorbed the most efficiently by our respiratory tract, like dust, right? When we expose to dust containing lead, we can actually ingest it. It goes to our body. Ingested is absorbed by small intestine and certain gastric juices in our stomach and so on. Right? So after oral ingestion, you have 60% in bone. Okay, this is the storage. Okay, lead storage is not desirable because it remains in our body. It goes to hair, it goes to teeth, and then 25% in liver, right? 4% in kidney and 3% intestinal wall. Endpoint would be sulfhydryl enzyme inhibition. As I mentioned, this is a group whereby it can bind with lead very easily. So you have inhibition, and then it can also inhibit this potassium transport in red blood cell, right? And it can cause a certain effect like anemia. Okay? If you have inhibition in your red blood cell, your blood will be affected. Okay, the ability of your blood to transfer oxygen will be limited, right? So we have porphyrinuria. Okay, you can also excrete certain proteins. Okay, it damages your kidney, and you can excrete it through your kidney. Okay, so chelating agent for addressing this kind of problem would be EDTA, penicillamine, and dimecaptol, or we call it as BAL. BAL is a name used for this dimecaptol chelating agent. Okay, so you can see here, right? We can actually remove it, this lead, okay, in our body through this chelating agent, right? Okay, one case study associated with this lead would be something called the illicitly distilled alcohol, or what we call as a moonshine, right? So lead poisoning is quite serious, right? So lead poisoning, if it goes to our body, we can actually carry out certain metabolism, right? We can actually excrete it from our body. But if it's excessive, then of course this lead will be distributed in our certain organ, which can cause poisoning. Okay, so it goes to a certain organ, for example, our liver, and then it causes inhibition of the enzyme. Okay, so for this case, we have this moonshine, okay, which is something that they use to distill alcohol, right? In the US, in 1991, there are many people who like to distill their own alcohol, right? Something that we shouldn't drink, right? Alcohol is something that can cause a lot of effect to our body. So what happens is that there are 8 people, okay, 8 person, they use this moonshine, they distill alcohol, and then they drink it. Okay, so they don't know that this alcohol that they distill is actually contaminated with something called lead. Okay, so they test out, and then they see that the blood contains a lot of lead, okay, at the elevated level. So a manifestation can happen through various means. For example, like microcytic anemia, right? For example, a blood problem. You have a lot of effects like extremity weakness and and so on. So many effects, right? So this is how it looks like when they distill the alcohol, right? So if they use something that contains a lot of lead, for example, when they distill it, then I think we have another example photo. No. Yeah, so when they distill it, if the container contains a lot of lead, then of course it will be exposed to it, right? So what they used was I think they used something from the car, right? Their car has maybe the radiator or something contains lead. So when they use it, then they start to have this lead effect, and of course the treatment will be the chelation therapy, whereby they are given certain chemicals that can compete with the receptors and enzyme for the lead. Okay, so in the end, I think everyone recovered. Okay. Sorry. Yeah. So that will be the metabolism part, whereby we talk about the phase 1 and phase 2 reaction. The next will be the elimination of toxicant. Okay, the last part of toxicokinetics, right? What we want to see in elimination is to see how our body can actually remove it entirely from our organ. Okay, so there are several ways, urinary ways, like using urine. Okay, we excrete it through urine. We have the fecal way. Fecal means that we go to the toilet, we remove it through our intestine, right? By feces, right? We have respiratory means. Okay, respiratory means that we breathe out. Okay, we breathe out certain chemicals, right? So other ways will be saliva, sweat, milk, okay, nails, hair, skins, and even the cerebrospinal fluids. So kidney would be the best way to remove the toxicant because kidney itself can remove water, right? Easily. It filters, okay, filters our certain chemicals in our body. It prevents those proteins, large molecules from going out. Okay, while those water-soluble toxicant can actually be excreted. Okay. So yeah, several ways whereby kidney can remove your water. In the function will be through this mechanism: glomerular filtration, tubular secretion, and tubular reabsorption. So all these are related to the so-called function of the kidney, which I don't want to cover. This one you can go through it to see how the kidney can actually function. But what you need to know is that the kidney itself is a main filtration system that can be used to filter off your toxicant, right? And then, of course, we have the fecal excretion. Fecal excretion means that we actually remove it through our feces, okay, feces. You have the exhale air, okay, the other one will be the exhale air as one of the so-called pathway to remove your toxicant. This is usually conducted through gas-phase xenobiotics, okay, those xenobiotics that can be in the gas. For example, carbon monoxide, right? Carbon monoxide you can breathe in carbon monoxide, and then to some extent we try to excrete it out through this exhale air. Okay, so this is usually through passive diffusion from blood to alveolus, okay, via a concentration gradient. It's because our body contains a lot of this alveolus, right, which is in contact with the external and internal part. Okay, the blood actually can release certain things out from the this lung, okay, and excrete it through exhale air. Okay. Okay, that's for our toxicokinetics. We have gone through certain things like absorption, distribution, metabolism, and excretion, right? So the next one would be looking into target organ toxicity. We'll look into how a certain toxicant can affect our different parts of our organ, right? Target organ toxicity means the adverse effect. Okay, adverse effect manifested in specific organ. We want to see how a toxicant can go into a certain receptor, and what is important in this target organ toxicity that can make a difference. Why certain chemicals can affect lung more than your heart, sometimes your kidney more than your liver. So we'll look into that. So one of the reason, one of the main factor that can cause this adverse effect due to the toxicant will be what we call as a cardiac output. In other words, we are talking about the blood flow, right? Blood flow in our body is very different, right? Certain chemicals is in our blood more. Okay, in our blood more means that. Okay, in our blood more means that, okay, let's say you have lead, right? Lead in our blood, right? If you have a lot of lead in our blood, then of course, generally it makes sense that if an organ receive more blood than other organ, then this particular organ that receive more blood will be exposed to more lead, right? So the cardiac output is an important factor that controls the exposure of the toxicity, right? And the other one would be the organ. Okay, organ has specific tissues, right? Specific tissues means that we have certain receptors, right? Different kind of tissues have different kind of cells, and different kind of cells have different receptors for specific functions. For example, your kidney has certain cells that allows filtration of the water, right? So these kind of cells, they are susceptible to certain toxicant. And the other one will be your liver. Your liver has a lot of enzyme, and these enzymes has a lot of functional groups that can be susceptible to certain reaction with a toxicant, right? So this is one of the reasons why certain organs have certain susceptibility to a certain pollutant, right? It's because of the difference in terms of one, the cardiac output or the blood flow. Two, in terms of the receptor itself, it has more receptors than other organs that is susceptible to that particular toxicant, right? So generally, toxicant, a particular chemical, don't affect all the organs, right? It depends on the characteristics of the toxicant itself. It may have several sites of action. It may affect certain organs. It may affect only one organ, right? But like mercury, it likes to attack the central nervous system, right? And benzene, of course, is likes to go through the carcinogenic pathway, right? And it depends on what kind of toxicant that you have, the characteristic of toxicant. So a multi-toxicant exposure may target the same organ. Okay, like I said, it depends on the characteristic of toxicant, which we'll look into in more detail next week on how a different kind of toxicant can affect certain organs and what is the characteristic of toxicant, right? So this is just to show you the cardiac output, which means the blood flow to the system. In the whole body, we have 100%, and then we have liver, which receive most of the blood flow, 27%. And then we have kidney, also receive a lot of blood. Okay, liver and kidney, they are very essential in detoxification. Liver is more towards the metabolism, right? Biotransformation. So it receives a lot of blood from the body, right? And then it also receives a lot of this blood from the intestine. So we have kidney as a filtration system. Kidney itself also receives a lot of blood. And then we have the brain and skeletal muscle. Skeletal muscle is 15.6%. But then, of course, when we are exercising, right, this 15% can actually increase to 60%, right? It depends on the work that we are doing, right? So so you can see that there's a difference in terms of the blood flow. So if you talk about exposure, right, if you have a toxicant in your body, the liver itself will be exposed to more toxicant compared to your kidney, brain, and so on, right? So blood circulation, example, you have circulation from all over the body. So the more blood you receive, the more exposure it is, right? So just to show you what kind of problem that we can have when we are exposed to toxicant, in terms of the toxicodynamic study, right? When we talk about toxicodynamic study, we want to look into what kind of toxic effect a particular toxicant can cause. So I'm going to share with you certain common toxicity, okay, that you can see. The first one will be the hemotoxicity, which means that it is the blood cell toxicity, right? Toxicosis due to chemicals that can lead to blood cell problem. Okay, so one example would be this hemotoxicity caused by several toxicants, for example, nitrate, cyanide, you have carbon monoxide, you have hydrogen sulfide, you have zinc. Okay, so all these can actually affect the normal function of the blood cell, like erythrocytes, red blood cell. Right, you have iron. Right, this iron can interchange from the Fe2 plus to Fe3 plus, and then to Fe2 plus. So it can actually interchange, right, in terms of the ability to accept, okay, oxygen and then release oxygen. So this iron itself has to change so that it can facilitate this kind of process, the interchange between oxygen. So if you have certain chemicals like nitrate, cyanide, okay, carbon monoxide, it can bind with certain parts of this erythrocyte, okay, or the red blood cell, right? It can bind with certain parts like iron part in our hemoglobin, right? Then it will cause this iron to not be able to bind with oxygen so effectively. In other words, it can impair our ability to carry out respiration, okay, to bind with the oxygen and to release oxygen. So it will cause our body to suffer from certain effects, like, for example, anemia. Okay, anemia is the condition whereby our body cannot have this oxygen transport. Okay, cannot have efficient oxygen transport due to the defects in our red blood cell. Okay, we have a leukemia, we have thrombocytopenia, we have granulocytopenia. All these effects are the red blood cell effects. Right? Of course, in terms of evaluation of the effects of red blood cell, we have the complete blood count, we have the ABG, arterial blood gas, we have evaluation in terms of toxicant or metabolite concentration in our blood, right? Okay, one case study related to this hemotoxicity would be this ingestion of sodium nitrite. Okay, sodium nitrite is one of the chemicals that can affect the ability of the red blood cell to facilitate the oxygen transfer, right? So if you have this sodium nitrite, okay, it can actually affects the ability of Fe2 plus and Fe3 plus in your cell. Okay, so we have this case whereby several adults, right, they reported certain effects like dizziness, you have lightheadedness and cyanosis, which means that the blood is actually blue in color. They observe blue in color in their hands, right? So they observe all these symptoms after they share a meal. Okay? So they suspect that the meal itself is a toxic meal because there's another person who did not eat the meal and did not suffer from anything. Okay? Just the 5 person who eat this meal starts to see certain problem. Right, and of course, the younger woman unresponsive. Probably this younger woman eat a lot of this thing, right? This meal usually for sodium nitrite, they use it for meat. Okay, they use it to process the meat, right? So this younger woman may probably eat a lot of this meat, okay, and then probably lost consciousness, right? And then some of them have to be intubated, right? So after a certain time, the healthcare person, they administer this methylene blue. Okay, they identify that these people suffers from certain blood problem. Okay, after eating that, they found that from the observation, they found that these few people, they have certain issues with their blood. And most likely from the food itself. So they administer this methylene blue. And this methylene blue can actually act as a reducing agent, right? it can actually be used to solve this issue, sodium nitrite issues. Luckily, all these people recovered. Right? So follow-up investigations show that this sodium nitrite is actually used for meat curing, and it was wrongly labeled as iodized table salt. So this caused the chef, okay, to cook it using this salt, okay, sodium nitrite, and unfortunately, it caused a lot of issues to human, okay. Okay, so the next manifestation will be the hepatotoxicity. This hepatotoxicity is due to the problem with your liver. Okay, so your liver itself is very important for biosynthesis, for nutrient metabolism, for toxicant metabolism, right? So sometimes when you ingest certain toxicant, your liver may not be able to detoxify it, right? Sometimes your liver may suffer from the effect, right, of the toxic effect, toxicant itself, right? So as you know, your liver has the highest cardiac output, which means that it has the highest so-called blood flow to the system. Okay, it is also having this something called enterohepatic recirculation. This is one of the nature's way for us to recirculate our nutrient, right? But of course, if you have toxicant in your blood and it undergoes this enterohepatic recirculation, you can have potential re-exposure to the toxicant itself. Okay, so if you have this hepatotoxicity, you may observe certain things in your liver. For example, for this case, you can see that your liver have certain inflammation, right? You can have necrosis, death of the liver cell. You can have scarring, okay, certain scarring in your liver cell. So you can see that there's a certain issues with this liver. And this is like bleeding, but this is a whiter part, okay? There's an inconsistent texture or morphology, right? So we can say that this liver is suffering from certain toxic effect, right? It is not a healthy liver. Okay, so your hepatotoxicity can manifest in certain ways. For example, lipid peroxidation. Okay, this is oxidation of liver cell. Okay, your liver cell may be oxidized undesirably by certain radicals, right? Like sometimes you ingest certain chemicals, right? That contains a lot of radicals, right? You eat it, and then after that, it can be metabolized into radicals, right? And it can actually oxidize your lipid, right? Your liver cell, okay? It can oxidize your liver cell, make it into something that is not functional. Then you have a necrosis, death of liver cell. You have fibrosis, which means that sometimes your liver can have scarring. You can have change in morphology of your liver, right? To become something that maybe like a fiber-like structure, which is not functional compared to the normal morphology. You can have a fatty liver. Fatty liver is a common problem, right? This is reversible, right? I have a lot of colleagues in USM who suffers from these fatty liver issues. But of course, with a healthy lifestyle, you can actually recover from this fatty liver problem. Aside from this, you may have a bowel interruption, right? You may have a certain problem with your liver, your bowel, right? Which affects the way you digest fats, right? Okay, so how to know whether liver function is impaired or inhibited? We actually test certain things, like for example, we actually look into the liver enzyme and sometimes gross tissue effects, right? Sometimes in your blood, right, you can actually have certain liver enzymes, right? So when your liver is damaged, let's say this is your liver and you have certain damage, right? The enzyme in your liver can actually leak out, okay, leak out into your blood, right? So when your enzyme leak out into your blood, okay, a simple blood test of the liver can actually tell whether your liver is working properly or it is actually having problem, right? So most of the time when you go to a body checkup, they will take your blood and then they will test for this kind of enzyme, right? And the other one will be to sample the liver itself, right? This is a very invasive way of looking into the liver health, right? There are people who needs to carry out this kind of test, whereby the doctor will actually take some of the liver tissues, okay, through maybe like a needle, right? Poke into your liver and take some of the tissues, and then after that, check what is the morphology, look into the whether it is normal or it is not functioning well. So this is what some of the tests that doctors can do, right? One of the problems that cause lipid peroxidation and the damage in your liver will be the oxidative stress, right? So oxidative stress means the free radicals generated, which can cause effects, like oxidation effects, right? Damage your DNA, affects the organelles of the cells, and so on, right? So some of these radicals will be like superoxide radicals, very, very common radicals. And this kind of radicals is very, very frequently generated in your liver, right? Of course, this is the reason why we need something called an antioxidant, right? Antioxidant can be enzyme, right? It can come from enzyme. For example, catalase, superoxide dismutase, glutathione peroxidase. So all these enzymes, they can act as antioxidant to remove the radicals, okay, from your body, right? Aside from this antioxidant enzyme, because from our food, we can also eat something that contains antioxidant like vitamin C, right? I don't know whether you heard of this marketing strategy. A lot of people like to sell something, okay, food that contains antioxidant. They will write something that it has a lot of antioxidants, right? So we need to take in this antioxidant because it can help us in removing these radicals from our body, right? They can actually react with this antioxidant can actually react with the radicals, okay, and effectively quench it, okay, quench all these radicals, right? So sometimes we have this compound, xenobiotic compounds that can be undergoing redox cycling. Okay? This kind of compounds can cause a lot of nasty things in our body, right? Sometimes this toxicant can facilitate the radical formation, and it is actually rechargeable, which means that these xenobiotic compounds, it can be oxidized, can be reduced continuously in our body, and in doing this process, it is actually creating a lot of these radicals, right? And this radical itself can lead to lipid peroxidation and also DNA breakage. In other words, it can cause cancer. So the antioxidant that you eat from the from like fruit, like vitamin C, all these, right, can actually help in decreasing the cancer risk, right? Because if you have a lot of these radicals, they can actually attack the DNA strands. Okay, when it attacks the DNA strands, it actually causes the DNA to be different. Okay, when it's different, it can translate certain information, right, to your cell that cause it to grow excessively. In other words, it becomes cancer cell, right? When it becomes cancer cell, then it starts to spread around your body, and then it can lead to a lot of issues, right? So redox cycling compounds, generally they are highly polar, right? And it can be oxidized by oxygen, and it can be reduced by flavin enzyme. Okay, there's an enzyme called flavin. Example would be paraquat, nitroaromatics, chelated metals, right? You can see from this cycle, your this is your xenobiotics, right? It can undergo oxidation and reduction, right? And in this cycle, redox cycle, it forms radical metabolites, which is something that will affect your body's health, right? Example, superoxide hydroxyl radicals, proxyl alkoxyl radicals for the reactive oxygen species. And for the reactive nitrogen species, you have NO, okay, oxides of nitrogen, right? They come from many sources, like breathing oxygen, UV radiation, infections, and sometimes oxidants. Sometimes the things that you eat may lead to the production of these radicals. Sometimes you eat something that is xenobiotics, human-made, and it can undergo this redox cycling, right? Then it will start to have a lot of these oxygen radicals coming out, and then you need to eat more antioxidant to remove those radicals. Okay. In terms of the characteristics, it is highly reactive. It initiates chain reactions, damage cells, cause degenerative diseases, right? In some people believe that these radicals, right, if it's excessive in our body, it can cause us to look older, right? Our aging process, all these are related to these radicals in our body. So if you eat a lot of antioxidants, right, you may look younger, right? That's what some people believe, right? And it's true to some point, because if you have oxidative stress or you have radicals in your body, it damages your cells, and it can actually make it age faster, right? So some people may have a body of like 20 years old, but their internal organs, their cells are already like maybe like 30 years old people, right? So some of the reasons is because of this free radicals effect. So antioxidants like vitamin C, beta carotenes, they are all able to quench the radicals, right? Even enzyme can actually quench it. But sometimes there are certain beneficial effects, okay, of this oxidation process. For example, if you need it for chemostasis, phagocytosis, which means that we want to destroy infected cells, or we want to destroy like bacteria and virus, this oxidation reaction can actually help in targeting that particular microorganism or bacteria to destroy it. Sometimes it can trigger clotting mechanism, which is good, prevent our blood from clotting. It can also be used for apoptosis, which means that it can actually be used for killing off our cell. Okay, sometimes our cell is not functioning, and sometimes the cell is not up to our expectation, our body's expectation, and we want to destroy it. So we can actually use these radicals. Our body can actually use certain mechanism that can lead to the destruction of the cell itself. So mechanism of action, decreasing ROS, RNS formation. Okay, if you take in antioxidants, you can actually decrease the ROS and RNS through quenching. Sometimes you have binding redox cycling metals, right? The redox compound can be eliminated. We have scavenging effect. We have adaptive antioxidant enzyme response. You can actually repair the oxidative damage and enhance enzyme repair. Okay, so example of antioxidants is something like this. All those fruits, they are very, very important for our health. Okay. Just to show you what exactly this redox cycling is and how it can generate radicals, and what kind of enzyme that can help in removing this kind of radicals. You can see here, this looks more complicated. If you have a parent compound, this usually is a xenobiotics. It can undergo redox cycle, oxidation reduction, facilitated by enzyme, flavin enzyme, right? And also this another enzyme, superoxide dimutase. So you can see in this redox compound, you can actually generate radical metabolites, right? And in doing so, you can actually also generate superoxide radicals from oxygen, right? So you have oxygen here, right? And flavin enzyme, as I mentioned just now, participate in this redox cycle, and it generates superoxide radicals, which is something that can cause oxidative stress. And then you have superoxide dismutase taking in this superoxide radicals, transforming into hydrogen peroxide, which can be further catalyzed into water and oxygen through catalyst, okay, and another enzyme. It can also be transformed into water through this GASH, right? And you have this iron in the body can actually be damaging, right? It can actually transform this peroxide radical into hydroxyl radical, which cause enzyme activation, lipid peroxidation, and DNA strand breaks. Okay, these are some of the endpoints. Endpoints here means that it is the manifestation of the problem or the toxicant, right? So oxidative stress has several endpoints like lipid peroxidation, DNA damage, enzyme inactivation, covalent binding to nucleic acids, and also covalent binding to proteins. Right. We also have this enzymatic response, which means that we have certain effects that can, certain tools in our body that can actually eliminate these kind of radicals. For example, you have manganese superoxide dismutase, which can change the superoxide into hydrogen peroxide. And then we have this selenium glutathione peroxidase, which can remove this hydrogen peroxide. Okay, this is not a radical, but it is also highly oxidative. It has a high oxidation power. So it can actually be transformed into water through several enzymes. Okay, this selenium is quite important. This is one of the reasons why we take selenium is because we have this enzyme which can be transformed into water, right? It can catalyze the H2O2 to water. Okay, so we have nephrotoxicity, the kidney toxicity, which if you look at the processes, we have these several processes that's happening in our kidney: glomerular filtration, tubular reabsorption, and tubular secretion. So all these processes helps to filter off those toxicant from our body, right? It helps to remove the water and while maintaining our body fluids. Okay, so when you have toxic effects, for example, some changes in this kidney, you can actually affect the filtration process. It affects the porosity, it affects the osmotic. So this kind of problem will actually affect the ability of the kidney itself to filter off proteins, okay, or certain things from our body. And if this happens, you'll observe proteins in our urine. So this is one of the tests in our hospitals, right? When you go to hospital, they ask us to get certain urines from our body so that they can inject it into certain machines, instruments, and look into whether our urine has proteins or not. So if our urine has proteins, this means that our kidney is actually failing, right? We actually not able to filter those larger molecule proteins from our body, right? So we have injuries, we can have certain problems like necrotic syndrome. We have certain toxicosis, like lead toxicosis, nephritic syndrome. All these, they are problems that we can observe in our bodies, our kidneys, like acute tubular necrosis, tubular reabsorption effects. And the other one is the more nasty one. The one that can cause a lot of pain will be this obstructive neuropathies, like for example, kidney stones. So this is one of the problems. A lot of times we heard a lot kidney stones in our body. So when we eat a lot of calciums and ligands, things like citrate, when we take in all these things into our body, sometimes they can go into our kidney and start to precipitate. Okay, they precipitate in as a stone, right? And then they can actually block the passage. For example, if you have a channel in your kidney, right, which transport water, and you have formation of kidney stone here, okay? So this actually blocks our water flow, and it will be very painful. Okay? When the water tries to push through this kidney stone, it can cause a lot of pain to your kidney. And to remove this kind of stone, you have to go through surgery. Okay, today we have quite a number of not so invasive surgery whereby they insert certain tubes, okay, into the kidney to break this into smaller pieces so that it can be flowed out from the kidney itself. A function indicator, how to know your kidney have problem, would be using glomerular filtration rate, active tubular secretion, removal of serum waste products, catabolism of proteins. For example, looking into whether you have detected creatinine in your urine, and see whether you have detected any muscle metabolism products in your urine. Then we have pulmonotoxicity, whereby I look into the gas exchange. Gas exchange means that we want to look into whether our lungs itself can function properly. So you know, like when we have COVID, if it attacks your lung will not be able to function effectively. You have a lot of fluids blocking your lung, and that will cause a lot of issues. Right, the endpoint here will include inflammation of the lung, death of the lung cells, carcinoma, or fibrosis. Right. Sometimes you have breathe in certain things like asbestos. It can actually stick to your lung cell, and then it can affect the morphology and the function of the lung. So just to go through this quickly, because this is not really related to the chemistry part, right? This is more on how this toxic substance can actually manifest in your body. Just to go through it quickly, like the more toxicity, this is irritation, okay, to your skin. Okay? Your skin is the largest organ in your body. Sometimes exposure to chemicals can actually cause irritation, like allergic reaction, phototoxicity, and sometimes you can have this integumentary cellular effects, like oops. I'm sorry, I'm taking a lot of time. I still have about 10 minutes, right? So I'm just quickly going through this. Yeah, dermotoxicity can actually affect the cell health. The last one will be neurotoxicity. Okay, this is one of the most important ones, right? This one would be damage to central nervous system or problem to your nerves cells. Okay, so if you look at your nerve cell, you have a lot of neurons. This one, I'm sure you have you know all this kind of morphology of the neuron. It looks very different compared to your normal cells. Okay, if you have certain damage to your neuron, you can actually affect the transport itself. So when you have this damage to the transport, you can affect the ability of the signal, okay, from one part to the other parts, from your brain to your target maybe organs, right? So when you have this kind of effect, you will have issues, okay? Issues with the signals itself. You will have a lot of manifestation in terms of your hand will start to shake, and then you may not be able to control your hand effectively. You cannot raise your hands, and because the signal from your brain cannot reach your target organ. Okay, sometimes you have paralysis, right? So I think I will stop here, and I will continue from this part next week. Just a short one, maybe like 5 or 10 minutes. Now I hope that I can take some questions. Hopefully the students will have some questions for me, right? Because I know that from last week, Dr. Fitri, you mentioned that some of them have a class, right, after this.
Yep.
So let's go through some questions to see whether, yeah, if you have any questions, just let me know.
All right.
Yeah.
All
so
right. Yeah, thank you very much, Dr. Wenda, for very comprehensive and detailed explanations, in particular regarding to the second last part of the toxicokinetic, yeah, regarding to the metabolism and excretion, and then followed by toxicodynamic. I guess already the questions from the student.
Oh yeah, I saw a question.
Yeah.
Let me read it out. I want to ask about xenobiotics. I don't understand what is xenobiotic exactly and how we study the xenobiotic metabolism. Okay, so maybe I did not mention what exactly is xenobiotic very clear. Basically, xenobiotic means man-made chemicals. Anything that we human made, right? For example, we have a lot of antibiotics, right? Antibiotics is considered as xenobiotic pollutant, xenobiotics compound, right? So we have like cosmetic products, that is also considered as xenobiotic compound. Anything that is man-made, okay, that's considered as xenobiotic compounds, right? How do we study the mechanism of xenobiotic metabolism? Okay, so it's not so easy to study the metabolism. So usually in clinical studies, right, we want to know how a particular chemicals, okay, can be metabolized in our body. We use animals, right? Animals like rats, right? Rats or maybe frogs, right? We inject these kind of chemicals into their body, right? And then through this kind of exposure, right, we determine, okay, for example, the first day we inject this chemical in the body. After 3 days, we take the blood from the animals, and then we determine what are the chemicals that comes from this xenobiotics metabolism, right? So we have a control study without exposure to these xenobiotics, and then we have animals that has been exposed. Then we look at the difference between the animal that has been exposed and animal that has not been exposed. We look at their composition of the blood, right? So if we determine certain chemicals that are present in animals that has been exposed to this chemical, but not in the one that has not been exposed, then we know that that particular chemicals is a metabolite of the toxicant, right? So we carry out this kind of studies to look at the comparison between the one that is exposed and not exposed. Then from there we can know. Okay, so should we have the data first about its effect on the body, so we can study its metabolism? Okay, generally you can have the data. Sometimes when we want to study about the metabolites, right, we not only study metabolites of xenobiotics, we also study metabolites of drugs, like something that is beneficial to our body. Right? We study many things, right? So some may have good effects to our body, some may have bad effect to our body, some may not have any effects at all. So this is an entirely different kind of areas. So if you want to study the effects to our body, we are actually talking about the dose-response relationship to look into how it can affect our body and what is the effects. The other one will be the metabolism part, whereby we look into what is the metabolites. So these are an entirely different field, and it depends on the objective of your study. You want to look at the effects or you want to look at the metabolites. So it's quite different. Can we study the xenobiotics metabolism through computational to see its prediction effect to our body? Yes, yes. There are people who looked into the metabolism using computational methods. But in the end of the day, right, computational study is just about predicting the structure. It is not really something that is like exact or certain. Regardless of whether you're using computational study or any other prediction study, right? The best one would be to get the real experimental data, which means that we need to have the real data that reflects how a particular xenobiotics is transformed into the metabolites. So I can answer you that yes, there are many different kind of computational studies using different kind of softwares, right? To look into the metabolism part, right? They use many ways to look into how an enzyme can actually metabolize certain molecules. Some people use DFT methods, right? There are many different methods, initial methods. Some use empirical methods, but then this is quite an emerging field, especially in the pharmaceutical industry, whereby they look into this xenobiotic metabolism using computational methods. I hope I answer your questions, right? Yeah.
Yeah, very great question from Adani.
Yeah, it's actually a very good question to look into this. Yeah, it also remind me that I did not really say what exactly is xenobiotics. Just now I did not define xenobiotics, right?
Yeah.
So yeah, I hope that can help the student understand better. It's actually a man-made chemicals. Right?
Yeah. All right. Still any other question? We still have 5 minutes more for questions. All right. I have one question, Dr. Wenda.
Yeah.
So on the metabolism part, especially on the biotransformation, you mentioned there are two ways, yeah? It can be goes to the phase 1 or phase 2 biotransformation. So on which or conditions or parameters or characteristic of the toxicant that it can be directly goes from the phase 1 and directly elimination, or from the phase 1 continue to the phase 2 and then goes to the elimination. So what kind of the characteristics, yeah, for the toxicant?
Yeah, usually for this kind of phase 1 and phase 2, right, we look at whether that particular molecule is polar enough to be excreted through our kidney. So sometimes when you ingest a toxicant, it goes to it is let's say it's hydrophobic, goes to our liver, and then it doesn't stop in our liver. You know, it will be undergoing phase 1 metabolism into something that has, like, for example, OH group, right? It becomes something that is soluble, and then it will circulate in our body. Okay, it will still go around our body, and then if it is able to be excreted by our kidney, then it will be excreted, and then if it's not excreted properly by our kidney, it will still go back to the liver. Because if you look at our body, our blood is circulating, right? So it doesn't just stop in the liver, and then after that, you have the phase 1, phase 2 there, and then only it goes to the kidney. It's actually going round and round and round through our body. So if, let's say, phase 1, after phase 1, and you still have this kind of pollutant or toxicant in our body, it goes back to the liver. Then after that, phase 2 will start to initiate, because our body cannot remove it effectively through kidney. So this will happen. Usually this is the case whereby a toxicant will need to undergo phase 2, because there's an accumulation of this phase 1 metabolites in our body, right? Yeah. So in terms of the functional groups, what kind of pollutants or toxicant in our body, that will depend. In fact, our body has evolved in such a way that it is not that selective anymore. It is able to react with different kind of products or compounds, right? Different kind of compounds, they are not so, sometimes they have like benzene ring, sometimes you have phenol, sometimes you have naphthalene, sometimes you have different kind of molecules, right? So our body has evolved in such a way that for us to remove these kind of chemicals, these classes of chemicals, we will have enzymes that can introduce OH group. We have enzymes that can introduce maybe like an epoxy group to make it into polar compounds, and then after that it can be secreted by our kidney. So yeah, this is I hope I answer your questions.
So it really depends on the hydrophilicity result, yeah, after the transformation.
Yeah, it depends on whether our body can actually remove it away, right? Yeah, our body can remove hydrophilic compound more effectively compared to hydrophobic one.
I see. And one regarding to the metal toxicity, you mentioned the good example of lead. Yeah, I ever read some references that sometimes there is also the typical mechanism, which is one time the metal toxicity occur in our body, it can causes a kind of mimicking mechanism. For example, lead very close to the cadmium or zinc or something, and then it can impair some functions, yeah, of that metal ion in our body. So could you please comment for that one?
Oh yeah, I agree with you on this. Yeah, this is one of the effects of the lead. But lead, generally from what I know is that there are many kind of manifestation of this toxic effect, toxicosis of this lead. So you can lead can mimic certain chemicals, certain heavy metals because of their similar characteristics. But in the end, how lead binds with those kind of proteins or maybe like enzyme will be based on, most of the time is based on this ligand to metal interactions. They form complex, right? So this is the one of the main way of lead binding and causing toxic effects. So it can mimic, okay, it mimics certain heavy metals, and the mimicking effect is also part of the ligand interactions, right? As far as I know, yeah. And the major toxic effect that comes from lead is, as what I mentioned, is through the inhibition of certain groups in the enzyme, which can cause the enzyme to be denatured, or in other words, not working properly.
All right. Thank you, Dr. Wenda. So to all students, if you still have any questions, quickly, we can, how to say, give you time for a few seconds, a few minutes. But just in case you have no other questions, maybe we can close this session, Dr. Wenda.
Okay, yeah, sure. So just to conclude, we have a assignment. Just to remind everyone that we have an assignment which needs to be submitted next week on the 7th of October, right? So this week we almost completed the toxicokinetics and toxicodynamics. This is the last part, neurotoxicity, which I will continue next week, right? Maybe like I will spend like 5 minutes to talk about this thing, and then we will move on to something that is more chemistry, right? We're going to look into the toxic effect of the pollutants, what's the characteristics of the pollutants, and how we can actually class them, and what exactly is the characteristics that can cause it to be more readily absorbed, and so on, right? So next week will be more on that, right? So that's all from me.
Yeah, thank you very much for the, how to say, reminder about the assignment, and also the next lecture will be delivered next week. And for the assignment submission, you already provide the link.
Oh yeah, you can go to the link, the one that I mentioned just now.
All right.
And then in the assignment itself, there's a link. Okay, that inside the assignment there's a link for the student to submit.
All right.
Maybe I can just quickly show, if you don't mind, just
please. We still have to.
Let me just unshare first. Just 1 minute. Let me...
No problem. Yeah, I also encourage to students to also resubmit to the, how to say, our learning management system. We call it SPOT, so you can submit to the both platform. First, the link that provided by Dr. Wenda, and secondly to the SPOT. This one, right?
So can see. Okay.
Great.
So this is the assignment. Assignment 1 is I put it as graded 15%, right? So you have a graded 15% for this assignment 1, and then presentation will be 50%, right? And then the other one will be the last lecture. We will have a 35% cognitive based questions. So these questions, you can see they are part A and part B. So part A will be very simple short questions, right? This is like you can refer to the notes, and then after that you can just give the answer very easily, right? Part B will be more on the question problem solving. I only give 3 questions. For now only 3 questions. This is more on the how we can actually solve some of the problems, and this one is based on the research. I call it as research question, whereby the students will need to carry out certain research. You need to go online, maybe search for journal articles, look into, for example, number 2, arsenic as one of the toxic heavy metals. You need to find out what is the sources, speciation, target organ, environmental concentration, right? You need to do some research. And then the third one will be to solve this problem. Okay, we have some data and how we can actually use this data to find out certain parameters.
All right.
Yeah. Okay.
All right. Thank you very much, Dr. Wenda, for briefly, explain about the assignment. I do hope the student can follow the guidance and then do your best to complete this assignment. And don't forget the submission due date will be next week on 7 October 2022. And please kindly submit your response on the link that provided by Dr. Wenda. All right, I guess we come to the end of the lecture. Again, Dr. Wenda, thank you very much for your great lectures. And yeah, I wish you a great weekend and very hope to see you again next week. Thank you so much. Wish you a great day, Dr. Wenda. Bye-bye.
You too.
Yeah, bye-bye. Thank you. So to all students, hari ini kita akan melanjutkan kuliah ya. Nanti silakan dicek di link yang diberikan untuk assignment-nya. Sangat komprehensif lecture-nya ya, more or less ya, kalau saya menyampaikan materi ini juga mungkin persis sama dengan apa yang disampaikan Dr. Wenda. Dan terima kasih, tadi ada nih pertanyaannya sangat baik ya. Yang lain juga kalau nanti ada pertanyaan silakan bisa disampaikan dalam bahasa Indonesia, nanti I can support you to translate in English. Berikutnya kita masih ada kuliah untuk analisis lingkungan. So, gimana baiknya? I would like to hear from you whether we conduct the lecture online or we move to the classroom at 9 o'clock something. Halo?
Online aja mungkin Ibu.
Online aja?
Ya.
Oke, kalau begitu kita break dulu sekitar 10-15 menit ya. Saya close linknya karena ini untuk report ke program WCU. Nanti saya berikan link berikutnya untuk analisis kimia lingkungan. Oke?
Baik Ibu.
Ya, terima kasih. Sampai berjumpa kembali sesaat lagi di jam 9. Lebih sekian. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Ibu.
Terima kasih banyak.